Good morning, everyone. Thanks for coming to this very special colloquium today in the Bren School. It's my great honor to welcome Professor Tyrone Hayes from UC Berkeley. Professor Hayes has come here because primarily of an invitation that's associated with a seminar in ecotoxicology in the Bren School, and we're very grateful for him coming here today. He was originally from Columbia, South Carolina. He did his bachelor's degree in biology at Harvard University in 1989, went to Berkeley for his PhD in integrative biology, and Berkeley snatched him up. He did his his assistant professorship, associate professorship, rapidly climbed through the ranks up to full professor, where he's been ever since, since 2003. His research primarily focuses on developmental endocrinology. He's done an extensive body of research on effects of atrazine on amphibians, which motivated this invitation primarily, so we're looking forward to hearing about that today. He's won numerous awards in teaching. He's a very devoted teacher, won awards in teaching and mentoring from UC Berkeley. He's won the Jennifer Altman Award for Integrity in Science, the Rachel Carson Memorial Award, the National Geographic Explorer Award, and from the American Institute of Biological Sciences, the Presidential Citation Award. I'd like to add an aside that this morning I had the great pleasure of watching Professor Hayes in action with a body of fifth graders at Harding Elementary School. So it just so happened that the students were studying his module on the frog scientist. The email asking if he would be available by phone conference coincided with our exchange about this invitation. Once that connection was made, the teachers there were elated that he could also combine his Q&A, and it was wonderful to hear both the questions and answers and to see the spirit in the room with Professor Hayes this morning, inspiring these fifth graders at Harding School to be excited about science. So I want to welcome you. Thank you for being here, and we'll be hearing from you about From Silent Spring to Silent Night, A Tale of Toads and Men.
So thank you very much. So, yes, if you've heard, I've had a pretty tough day already. Those fifth graders really don't let up, so nothing you can throw at me now will faze me. I want to thank you all. I want to thank you for the invitation. I want to thank you all for coming. And I'm going to spend the rest of my time explaining my title, From Silent Spring to Silent Night, A Tale of Toads and Men. Before I do that, as usual, I want to give out my acknowledgments and my thank yous to make sure we get the important stuff done. As always, to my wife and my son and my daughter for their love and support. And I also want to acknowledge, and as a matter of disclosure, acknowledge my funding sources. And disclosure in particular, I've been funded by Novartis Syngenta, the manufacturer of the main chemical I'll talk to you about today. And periodically, because they have a slight disagreement with my interpretation of the literature, I will insert what I call words from my former sponsor, just so you get sort of both sides of the argument. Next, I want to thank all of the students that have been involved in about 10 years worth of work that I'm going to talk to you about. And in particular, I want to point out that everybody in blue here was an undergraduate. I had a wonderful experience as an undergraduate myself, which had a huge impact on my career. And for the last 20 years, I've been trying to give back what was given to me. And then to my new crew, so these are my most current group of students. And as you can see, we enjoy quite a bit of diversity in my laboratory. And finally, to my family at large, where we also enjoy quite a bit of diversity, for their love and support as well. And this talk, as is all of my talks for the last 5 years, is dedicated to my grandmother, who passed away Christmas Day 5 years ago. Luckily, she passed on her love of teaching and her desire to make the world a better place to me before she herself passed on. So this whole story starts basically with a little boy who likes frogs, even before I was in fifth grade. The sort of professional entry into the work that I'm going to talk to you about today started with this guy. This is Hyperoleus argus, and we're having a slight problem with the color and projector, but this one should be bright green, and this one should be red with sort of white spots. But what's fascinating is that the male and female, even if the colors aren't correct, the male and female of the species look so different that you'd think they weren't in the same species if you saw them. So these were collected in the Arabuca Sokoke. And again, just as a little boy who likes frogs, I was interested in what made these frogs different colors like this. I brought some back to the laboratory, and this is the same frog photographed once a day for 6 days. And we discovered, or we found out, that they all start out green at metamorphosis, whether or not you're male or female. The females at sexual maturity, months down the road, would then change color to this sort of spotted pattern. We applied testosterone just by dipping these frogs in the water and showed that the so-called male hormone had no impact on their color change. But we found that we could dip these guys in estrogen-contaminated water and induce prematurely the female coloration, regardless of whether or not you were a male or a female. And those are the kinds of simple, not useful kind of questions that I was interested in. Just a little boy who liked frogs. And then I'll never forget, about 18 years ago, and I remember the date for the following reason. I was giving a talk at UC Davis, and my wife, that beautiful woman I showed you earlier, was in the audience, and she started to have contractions. My first and only son was on the way. And while we're driving back down the highway, she says, she has an MBA and an MPH. She says, you should patent that. And I said, what do you mean I should patent that? And she calls up her brother-in-law. He says, oh, you can patent a frog. Heretofore to be referred to. See, he's a lawyer. And so we did. We called this thing the Hyperoles argus, that's the species, endocrine screen, or the Hayes test. And you might be asking yourself, why on earth would anybody patent a frog? And again, I never thought I would do anything useful in my career. Well, it turns out that this is a control, an unexposed animal. They're about the size of your pinky nail, which I guess depends on the size of your pinky. But if we dip these guys in estradiol, the natural steroidal estrogen is present in everything from fish to farmers, we can induce the pattern color change. If we put them in ethinyl estradiol, this is the estrogen that's used in birth control pill, we can induce this color change. If we dip them in diethylstilbestrol, a non-steroidal synthetic estrogen, we can induce this color change. And if you dip them in DDT, not a hormone, but a pesticide, an insecticide that can bind the estrogen receptor, we get a color change. It turns out that we screened dozens of compounds, and every time we got a hit, every time we got a compound that caused this estrogen-dependent color change, it was with a chemical that was also known to promote breast cancer, which is estrogen-dependent. So there was the usefulness. We had a little frog that we could raise by the tens of thousands in the laboratory, and we could screen compounds and ask, is this a potential breast cancer promoting compound? Or you could even send me a sample of your water and go, is there anything estrogenic in my water I should be worried about? And we could dip our frog in it and give you an answer. What's more is we showed that if we used the estrogen blocker tamoxifen, we could prevent the effect of estrogen. And at the time, this was a major treatment for breast cancer because it blocked the estrogen receptor. So we could even potentially screen compounds that might be anti-estrogens that might be used as pharmaceuticals for treating things like breast cancer. So we went on. We thought, oh yeah, we'll start a little mom and pop shop, have a little lab on the side and do this kind of work. And of course the university said, hey, that's our intellectual property and you have to show efficacy. We had to find somebody who'd be willing to pay for this little thing we had. So along comes a little company named Novartis. And they said, we have this compound atrazine we'd like for you to look at. I had never heard of atrazine. Now you Google atrazine, I pop up, you Google me, atrazine comes up. It's a so-called S-chlorotriazine. We're joined at the carbon bond. It's a so-called S-chlorotriazine. It's an herbicide or a weed killer mostly used on corn in this country, 90% of it or something like that. It's been used since 1958, so it's been around for a while. And we use 80 million pounds annually in the United States. I don't know if you can fathom 80 million pounds. It's a global problem. It's used in more than 80 countries, but now outlawed, or as the lawyers like me to say, denied regulatory approval in all of Europe. What's important about that is that the company is based in Europe. So keep in mind as we talk for the next 40 minutes or so, that we're using 80 million pounds of a compound that's outlawed on the continent that the company calls home. So once we got involved with this company, they said, well, we want you to use the African clawed frog, Xenopus, because that's kind of the well-known lab rat of the amphibian world. How many people know why that is? Turns out that in 1920, somebody discovered that the human pregnancy hormone will make this frog lay eggs. So by 1940, if you thought you were pregnant, you would go to the doctor and he would take a sample of your urine and inject it in the frog, and if the frog laid eggs, you were either happy or sad, depending on your situation. I introduce this way for a couple of reasons. One is I just get such a thrill out of wondering who was the first guy who thought, gee, I wonder what will happen if I inject pee into a frog. Like, how do you apply for NSF to even try that out? Things were different back then. The other reason I point it out is for the following. Just like with my hyperolase argus, the hormones, in case you don't know this, the hormones that we use are so similar, and in the case of estrogen, identical, that they will affect frogs and vice versa. The hormone that maintains our pregnancies, like the one my wife was ending as we came from UC Davis, is so similar that if I inject it into this frog, it'll make it lay eggs. So I want you to keep in mind in the first half of the talk, while we're talking about frogs, we're not just talking about frogs, that's the title of Toads and Men, we're talking about vertebrate animals in general. So the first thing we discovered, this is supposed to be red, sorry, is that atrazine inhibited growth of the voice box or the larynx in males. Now that's sort of a bad flag because the larynx in frogs, or the voice box in frogs, grows in response to testosterone or androgens, and it's the same reason that men have deeper voices than women. So the idea here was that we were somehow limiting the androgen production or availability in these animals so that their voice box wasn't developing. And if you're like a multi-billion-dollar company, that kind of makes you get concerned. Here are the reasons why. So here are the kidneys and gonads from an animal that we've dissected that's been exposed to atrazine as a tadpole. And this particular animal, this is from a paper we published in PNAS, has 2 testes, 2 ovaries, 1 large testis, more ovaries, which might seem like a party, but it's, oh, we're missing. That says not normal. That's what that says. And I point that out for several reasons, primarily because in the years that I've been talking about this work, somebody inevitably asked, isn't that normal for frogs? And finally somebody told me why. Who knows? Jurassic Park, yes. So apparently frog DNA in Jurassic Park made the frogs change sex, but that is science fiction. There are fish that are naturally hermaphroditic, but no such thing in frogs. So we made this proposal back then that the following mechanism might underlie this effect. So if you imagine that this ellipse is your testicle, I would advise you to imagine a friend's testicle, even somebody you don't like, because you should make testosterone if you have testicles. Testes. This word literally means testicular hormone. It is the male hormone. And our proposal was that when you're exposed to atrazine, it turns on the enzyme aromatase, the machinery, if you will, for converting testosterone into estrogen, a word that literally means generator of estrus, the female hormone, which is fine if you're a female. But if you're a male, the consequences are that you're demasculinized, because you're using up your testosterone. It's a little more complicated than that, but that's the main idea. And you're subsequently feminized because now you're making the female hormone, which if you're a male, you shouldn't be. So we were able to show in this first paper that indeed here's blood levels of testosterone in exposed African clawed frogs as adults. Here's the atrazine-treated males and the control females. So indeed, they were demasculinized, or as I like to say, chemically castrated, because the company doesn't like me to say that. And then we published a paper, Hermaphroditic Demasculinized Frogs After Exposure to the Herbicide Atrazine at Low Ecologically Relevant Doses. I imagined to get as many industry-offensive words in that title as I could. Proceedings in the National Academy of Science. And also, as I like to point out, there was 4 undergraduates there and 4 black men on one paper in PNAS and a Latina. That's got to be some kind of record. I'm sure we broke some kind of record. Despite that, 2 big questions were left unanswered. Are the hermaphrodites males with ovaries or females with testes? Which sounds like a kind of a weird question. But frogs don't have distinguishable chromosomes typically, and at the time there were no molecular markers. So we didn't know if we had males growing ovaries or females growing testes. And second, we didn't know what happened when these animals became adults. Do they remain hermaphrodites? Do they have both eggs and sperm? Do they become females? Do they become males? And in part, this was sort of an easy question to answer, but the difficulty was that they take 4 years to grow up. And even as an undergraduate or a graduate, you're only there for 4 or 5 years, so you've got to tell somebody, well, I have a project for you. And maybe in 4 years, you'll get some interesting data. And in fact, it never worked like that. The project passed on through several people over the last 4 years. And just this year, we finally published the results. And here's the answer. See, that guy kind of looks like he's smiling. As you'll see, we published this in PNAS. They said it was too racy for the cover. They wouldn't give us the cover. They put it on that little inside one, but they wouldn't put it on because that's his brother. So it turns out now we have a molecular marker. We can tell males from females. Females have this unique gene called DMW. And so we can genotype that guy, we can genotype that guy, and he's mating with another male. But it turns out he's a genetic male, but he's a female with ovaries that even lays eggs. We now have 3 generations of frogs that hasn't had a real female in it, a genetic female, for 3 generations. So 10% of the animals in this particular lineage completely converted into females, even though they're missing the female chromosome and the genes to make them a female. Before we published the paper, though, I said, okay, well, 10% of these exposed animals turn into females. The other 90% can't simply be normal. There has to be something more to this story. So we decided to look at fertility. We did some behavior work that I won't have time to talk about. And we showed, though, that the control males can fertilize about 85% of a female's eggs. Females lay about 2,000 eggs, which really impressed 5th graders, by the way. Atrazine-treated males, though, and that's supposed to be red, atrazine-treated males only fertilize about 15%. They have severely reduced fertility. And it turns out the reasons for this are a couple. One is they don't show mating behaviors. Mostly they just sit there and watch the females lay eggs like nothing's going on. But the other one is if you look at the testes under the microscope, the control animals, and if I blow this section up, here's a testicular tubule. All that dark stuff in there is sperm. Millions of sperm ready to go, sperm heads all lined up. In the atrazine-treated animals, here's a testicular tubule. Most of their tubules are empty and full of only cellular debris. So they don't make enough testosterone to properly make sperm, contributing to their low fertility rate. So then we were satisfied. We published another paper, again in PNAS. Atrazine induces complete feminization and chemical castration in male African clawed frogs. So I managed to pack a lot of good words in that title as well. And these are mostly undergraduates and 2 graduate students that were involved in that work. Now it gets weird. I have a student knock my win working on a project where we decided, because it turns out that different lineages or families, if you will, have different susceptibilities. And some of them, 45% of the males turn into females, and some of them 10, some of them 0. And we're trying to work out whether or not you can select for that and what's regulating that susceptibility. And so we set up all these different lineages where we knew the parentage and things. And Mai would come upstairs every morning and she'd say, I have to pull them apart. I said, every morning I have to pull. I said, what do you mean you have to pull them apart? Because I knew they were all male in these tanks, 40 males per tub. And so it turns out she brought me a photograph. And then we went downstairs to our animal facility, and sometimes you get things like this. That's quite a party going on. So then I said, well, that's not enough to have pictures, because I'm not a behavioral biologist. I said, we need some data. So she started going down now at 7 a.m. when the lights come on, and she would count. So if you look at that, that's about 7 pairs. And then there's the next day, and then there's the next day, and then, see, sometimes it happens in controls. So there's a background incidence of same-sex pairs or homosexuality, but it's a very different thing, okay? And these guys, it's random males just kind of hooking up. So we pit tagged them. We put in electronic identification tags. For these guys, it's the same males on the bottom every single time. There's about 12 males in this particular tub out of the 40 that are always in the female position or the female role. Now, one of the things might come to your mind is, in these all-male communities, that maybe it's big males overpowering little males. That's a possibility. So we looked at that. It turns out, if you look at, and this is just on one day's analysis, the ones on the bottom are always bigger, just the opposite, which is interesting because in the species, normally females are bigger. The ones on the bottom are always bigger, so it's not big guys overpowering little guys. If you look at their testosterone levels, the ones on the top are on average and almost always higher than the ones on the bottom. If you look at estrogen levels, which males shouldn't express at all, the ones on the bottom are making estrogen as if they're females. So what we think's happened is that a subset of these guys have become partially feminized so that they think because they have testes, but they think that they're females. They're behaving, their identity, as if they're female, and now we're working with a colleague, George Bentley, to describe their brain morphology because there are differences between male and female brains that control these reproductive behaviors. In fact, we have evidence that they're partially feminized in the following. These are testes, and this is one of these animals. See all that? That's oviduct. That's the equivalent of a man with a uterus. So they're still male, but they're partially feminized, at least in their secondary sex characters and in their behavior. The company's going to love that one. So the next thing we wanted to do, and actually we had to step back a few years, is we wanted to ask some questions about how widespread this phenomenon was. For example, African clawed frogs, I will admit it too, are weird. And so we looked at North American leopard frogs, and we showed that, so these are testes, and we showed that these guys don't become hermaphrodites, but they grow eggs in their testes. So this, what I call junk in the trunk, these are eggs that are yolking up in this male's testes and bursting through the surface of this male's testes. And this is work we published in Nature a few years ago. And in fact, by this time, I was in contact with the Environmental Protection Agency. And so I sent them, before this was even published, I sent them the data. I said, look what atrazine does to North American leopard frog. And they said, well, that's an interesting finding. But we're not sure if it's an adverse effect or not. And I go back to that time when I was driving home from Davis, and we ended up at Alta Bates Hospital, and my wife had that G.I. Joe Kung Fu grip on my hand, and she tells me, no matter what, you will never understand the pain of childbirth, and I can't argue with that. I would guess, though, that a dozen chicken eggs in my testicle would probably fall somewhere in the top 10. But the EPA doesn't recognize it as an adverse effect. So the next question was, is this a laboratory artifact, or can we find this kind of phenomenon in the laboratory? And just to give you an idea of what we're talking about, applicators, farmers, recommended application rates of 2.9 to 29 million parts per billion. We're getting these effects at 0.1 parts per billion. So a typical farmer might be applying this stuff at levels that are 290 million times what we're using in the laboratory. That results in agricultural runoff that can range anywhere from the low parts per billion range up to 10,000 parts per billion. That's micrograms per liter. Temporary pools look like this, permanent water, rainwater. Here's what it takes to make a hermaphrodite. Here's what on my screen is a red area showing you all the bodies of water that would be at risk based on our findings in the laboratory that these effects occur at 0.1 part per billion. Probably most impressive is that there's enough atrazine in rainwater to chemically castrate and make hermaphroditic frogs, up to 100 times. Turns out that 500,000 pounds of atrazine come down into rainwater in the United States every year, and it can travel for over 600 miles. The U.S. Geological Survey can measure it in rainwater in Minnesota from when they're applied in Kansas. It goes up on the dust, travels in the clouds. They've shown the same thing in Europe. It can travel for over 600 miles. 1.2 million pounds come down the Mississippi and go into the Gulf of Mexico every year. And the drinking water standard is 3 parts per billion, 30 times what we know to be biologically active. And that doesn't mean if you go home tonight, your water will be 3 parts per billion. That means on average over the year it's going to be 3. It might be 200 tonight. In fact, when I first started doing these studies, EH&S, Environmental Health and Safety, wrote me and said, well, we have some concern. What are you going to do with all that wastewater? And I emailed back and said, well, I'm going to take it home and drink it. Because it's guaranteed to, see, I thought it was funny too. But the EH&S boys don't play around. I think you get the point. At any rate, we did this. We went off into the field, and if you look at animals collected in the field, and now I've sliced out a section there, I'll blow that up. And what we find in the frogs in the field is that here's testicular tubules. There's oocytes, or eggs, instead of sperm throughout their testes. We tested this across the United States, and what we found was every place that we found atrazine contamination, we found these animals with testicular oocytes or vice versa. What I usually tell people too is that we controlled for latitude, but of course this is Highway I-80 and we were driving to a conference in Indiana, and so we got a nature paper out of the road trip. There we go. Now we're doing something a little more vigorous, I think. Instead of following the road, we're following rivers, so we've sampled over 10 years from the North Platte, the Missouri, and the Mississippi, so we can look at both spatial and temporal gradients and the intensity of sexual abnormalities as well as some immune function parameters that we're looking at. And that's work that we're working on publishing. I almost said by the end of the summer. I guess we missed that deadline. By the end of the semester. Then, and this is sort of the last little piece on frogs. we've been doing a few things to look now at pesticide mixtures, because you don't just get atrazine alone, but also to look at other environmental factors that might enhance or maybe even alleviate some of the effects of these pesticides. And we're looking at a broad set of developmental and physiological parameters. Salinas is one place we're looking at heavy pesticide use. So for those of you who don't know, 85% of our lettuce comes out of Salinas. And we look at a site right here, which I was telling somebody early this morning, in addition to all the pesticides, apparently people who don't want their puppies throw them off of this overpass. So it's a really nasty place to work. The point is, though, is that we're showing that there's a number of interactions that are contributing to amphibian declines. One is, you might imagine, as temperatures go up and ponds dry up and tadpoles become crowded, they show this stress response. Now, that could have nothing to do with us, or global climate change, draining water off for agricultural purposes may contribute. As the ponds dry up, there's an interaction. The pesticides are left behind and become more concentrated. And we've now shown that pesticides increase stress hormone or CORT levels. Those CORT levels contribute to the release of fat-soluble pesticides, increasing the internal loads of pesticides even more. That increases the stress response, which causes damage to the thymus and other aspects of the immune system. And we published a paper in Environmental Health Perspectives showing that in the lab, this leads to flavobacterial meningitis susceptibility. In the field, this causes an increase in pesticides that typically damage the kidney and liver in exposed animals. And the important part about these interactions, well, there's several. One is, if we didn't have the laboratory data to understand the role that pesticides play, then we would think that the major source of decline was just the pathogens and other environmental factors that the animals were experiencing. The other one is that the parasites, the trematodes that typically damage the kidney and the liver, effectively increase the pesticide loads, because when your liver looks like this, these nodules are all cestodes, you can't metabolize these compounds, and when your kidney's filled with trematodes, you have a limited ability to secrete the compounds, even if you could metabolize them. Just to show you that I'm well-rounded, I'd like to read to you a passage from John Steinbeck's East of Eden, published in 1952. So remember that work I just told you about in Salinas. In 1952, Steinbeck wrote, "Salinas was surrounded and penetrated with swamps, with tool-filled ponds, and every pond spawned thousands of frogs. With the evening, the air was so full of this song that it was a kind of roaring silence. It was a veil, a background, and a sudden disappearance, as after a clap of thunder, was a shocking thing. It is possible if in the night the frog sound should have stopped, everyone in Salinas would have awakened, feeling that there was a great noise. In their millions, the frog song seemed to have a beat and a cadence, and perhaps it is the ear's function to do this, just as it is the eye's business to make stars twinkle." I knew nothing about Steinbeck's writing when I first started working in Salinas, but given that description from 1952, I want you to hear now what Salinas sounds like with regards to native frogs. We haven't heard a single native frog in Salinas in the 5 years that we've been working there. And I believe that the pesticides have greatly contributed to the loss of the frogs in that area. We published a paper recently in Journal of Experimental Biology where we showed that clearly environmental pollutants are important, but they're interacting in an incredibly complex way with pathogens, atmospheric change, habitat loss and modification, and invasive species to contribute. And there might be one factor in certain areas, but we think in general we're going to get much further by looking at these complex interactions. I'm going to read this one. This is words from my former sponsor. "Earth Day," Tim Pasteur said, "is a good time to recognize the vital role the herbicide atrazine plays in protecting the environment and promoting responsible land stewardship." I want to sort of move on now because to get to the of toads and men part. Because what I think is interesting, having worked in East Africa, in rural East Africa, is that we're missing, with all of our fancy EPA and other agencies, I think we're missing something very basic that can be seen here very clearly, I think. This is Lake Nabugabo in Uganda, where the runoff from this agricultural field, which goes into these containers, is the sole source of cooking, drinking, and bathing water for this village. And what I think is interesting is I'm pretty sure if I told one of these guys in the village, the frogs in this pond are growing eggs in their testes and they have no sperm and their immune system's all screwed up, that the environmental health, public health connection would just be clear, a no-brainer. But I think because we have these agencies and our water comes out of this faucet and we don't see where it comes from unless we're flying over and we happen to know where our reservoir is, we think that there can't possibly be anything bad in our water. The title I use, From Silent Spring to Silent Night, is a play on Rachel Carson's Silent Spring, where she warned that the loss of birds due to pesticide exposure and our pending silent spring at the time was a warning to us, environmental health, public health connection. The global amphibian decline, where more than 70% of all amphibians are in some state of decline right now, the greatest loss in species since the dinosaurs, that I very much believe that this, our pending silent night, is also a warning to us, another environmental health, public health connection. Glenn Fox wrote, in ecoepidemiology, the occurrence of an association in more than one species and species population is very strong evidence for causation. I've told you that these effects occur in multiple species, genera, and even families of amphibians, and at least 5 other laboratories have shown similar things in amphibians. But there are also studies in fish, birds and reptiles, and even mammals showing that via many of the same mechanisms that we're seeing in frogs are affecting probably us as well. And I'd like to review those, a large part of which is not data from my laboratory, but data that I've now gathered from at least 40 other authors from around the world. The first thing I discovered when putting together a paper now that we just submitted is that here are my frogs. Remember the densely packed testis with sperm compared to the atrazine-treated animals, which I thought I discovered? Turns out a guy in Europe showed something very similar in fish, Kestemon et al. Here's caiman, so reptiles. So again, you can see densely packed testis almost looks identical to mine. And 2 laboratories have shown in rats. So again, you can see a densely packed testicular tubule with sperm, and that's what these rats look like when they're exposed to atrazine. So not multiple species or multiple populations, but every vertebrate class, except birds, are showing similar effects when you're exposed to atrazine. Is this due to a decrease in testosterone, which decreases sperm production? Probably. If you look in, and again, here's fish, work done in Europe, not my work. You see atrazine has a similar effect. Here's our frog work, and here's in rats, showing that atrazine significantly reduces testosterone levels. So it's not just something that some crazy, as the company would like for you to believe, not just something that some crazy guy from Berkeley has shown, but around the world in multiple vertebrate classes, independently we're seeing the same kinds of effects. And at least for frogs and fish, at similar so-called low doses. In humans, Shauna Swan showed that if you look at controlled men and compare them to subfertile men, that men who have low sperm count and trouble getting their wives pregnant have significantly more atrazine in their urine. And I don't know what it means, but these guys have about 0.1 parts per billion, about what it takes to chemically castrate a frog. If you now reduce the axes and look at Lucas et al. Works from 1993, here's the atrazine levels in field workers. And now, see, our data's still there. Here's the atrazine levels of men who apply atrazine in California, 2,400 parts per billion. So men who apply atrazine have 24,000 times the atrazine than we know is associated with low fertility in men in Columbia, Missouri. Men who apply atrazine have 24,000 times the atrazine than we use in our laboratory to chemically castrate and make hermaphroditic frogs. Or as I tell my so-called lay audiences, to put this in perspective, one of these guys could pee in a bucket. I could dilute it 24,000 times and use the atrazine in their urine to chemically castrate and make hermaphrodites out of 24,000 buckets of 30 tadpoles each. Of course, we know nothing about these men because they're primarily Latino, many with life expectancies of 50. So this becomes an environmental justice, an issue of environmental racism. In particular, when you consider California, that I don't know what we are now, but at least at the time I made this slide, we were the 5th largest economy in the world. Meaning that if we were our own country, we'd have the 5th largest economy in the world. 1 in 10 jobs are in agriculture. 30% of the land is in agriculture. We make 350 agricultural products. 50% of the United States' food, I didn't know this, comes from California. 50% of the United States' food comes from California. We use more pesticides than any other state, and 90% of the workers are Hispanic. So now if you consider all that, and we block out now the top 10 counties for agriculture, these are the counties that make us the 5th largest economy in the world. Now what do you think will happen if I plot the 30 poorest towns in California on this map? And then there's the 11th. So people who are being exposed to chemicals, including not just atrazine, chemicals like chlorpicrin, which was originally developed as a nerve gas in World War II. The poorest people in the state are making us the 5th largest economy in the world while faced with exposure occupationally to compounds such as atrazine and the others that I showed you. Words from my former sponsor. So the other half of the equation is, does atrazine turn on aromatase, leading to production of estrogen that in frogs causes vitellogenesis and oogenesis, the production of eggs, but in mammals, we know that estrogen is associated with mammary tumors and prostate cancer. What is the evidence? For prostate cancer, I'm going to read to you what they published in the International Journal of Occupational and Environmental Health, because the lawyer who keeps writing my dean claims that I'm misrepresenting and misinterpreting what they wrote in their publication. So I'm going to read to you what they wrote from page 1052. They wrote, quote, "The increase in all cancers combined seen in the overall study group was concentrated in the company employee group." Now, these are data that they collected in their manufacturing facility in San Gabriel, Louisiana, a community that's 80% African American, where they produce their atrazine. They wrote, quote, "The increase in prostate cancer in male subject was concentrated in company employees." Sounds like a nice place to work. They wrote, quote, "The prostate cancer increase was further concentrated in actively working company employees." So if you actually go to work, you're more likely to develop prostate cancer. They wrote, quote, "All but one of the cases occurred in men with 10 or more years since hire." So the more loyal you are to the company, the more likely you are to develop prostate cancer. And finally, they wrote, "Analyses restricted to company employees also found that the prostate cancer increase was limited to men under 60 years of age." Now, prostate cancer is something you get when you're over 65. All but one of these men were 50 years old, and the increase was 8.4-fold. They claim it's because they screen their people better, but this is compared to men who worked in the factory but didn't work in the room where they bagged the atrazine. With regards to breast cancer, at least one study has shown with a p-value less than 0.0001 that women whose well water is contaminated with atrazine are more likely to develop breast cancer as compared to women who live in the same community but don't use the well water as their drinking water. Correlation, however, as you know, despite what the p-value might be, does not tell you causation. But we have a rat model where we've shown that atrazine decreases testosterone, and there's a concomitant increase in estrogen, which we know promotes breast cancer. This work was done in an EPA laboratory and published. What's more is, Syngenta's own laboratory published a paper showing that atrazine increases the incidence of mammary tumors, and another paper in Japan showed that those mammary tumors are estrogen receptor positive. So in a rodent model, we have a reasonable mechanism that will support the correlative studies. What's more is, it's not just a rodent model. If you look at human cancer cell lines, and in this case it's an adrenal carcinoma, this is background set to 1, they don't make aromatase, they don't make estrogen, but if you give them atrazine, they express aromatase and start making estrogen, just like we've shown in fish, just like we've shown in frogs, just like they've shown in rats. Atrazine induces aromatase and increases the estrogen-androgen ratios. And this is work that came out of Europe. I visited them. They wouldn't let me in, though. In San Gabriel, Louisiana. In fact, I was just there 2 weeks ago for something else. They have a pipe that goes right into the Mississippi that's letting something out. I don't know what it is. Most of it looks like that in the low-income black community where their factory is located, which, by the way, their lawyers wrote a nice letter saying that they don't put their factory there because there are black people there. They put their factory there because it's cheap land, which just happens to be where black. I said, really? That's your argument? Here's why I make the point that it's an African-American community. These are the top 13 cancers that people get in the United States. And you're not going to see this now because we're having a color issue. But in red, all but these two are all the cancers that African Americans are more likely to get relative to Caucasians. What I'm showing you on this graph now, normalized to 1, are now all the cancers that black people are more likely to die from relative to Caucasian Americans. And so the question is, are there really, and I can show you a similar map for Hispanic or Latin American, Latinos. The real question is, are there real biological differences that contribute to these racial disparities, or is it because as minorities we are more likely to live in communities and work in occupations that would increase our exposure to chemicals that we know are associated with the kinds of diseases such as cancers that we're talking about? One of the things that we've been working for in Berkeley right now is to try and answer these questions, but there are not even any cell lines available to address the issue. All of the cancer work we do on breast cancer right now, of all the money we spend, none of it is done on cancer cells that are relevant to the people who are more likely to die from those very diseases. And so we can't even start to answer those questions. So again, I'm just a little boy who likes frogs, but what excites me now is that this work that I've done in this aquatic organism is really starting to tell us, I think, something about this aquatic organism. Because indeed, this is when we're most vulnerable. Even though as adults, we can metabolize it and urinate out the atrazine, these little guys are no different than my tadpoles in a bucket. They might be able to metabolize and urinate it out, but they're in the same water, and they just resorb it over and over and over again. In fact, though, we've used this aquatic organism to help us out with the one that I was initially interested in. We've used human cell lines now, me and colleagues from Japan, to show that not only does atrazine, and I won't make you walk through the whole mechanism, not only does atrazine bind into cytoplasm, contributing to increased expression of aromatase, but we've shown that it interacts with a transcription factor in human cell lines that contributes to the overexpression of aromatase in human cell lines. And aromatase, the promoter in frogs and fish and dogs and cats and hogs and lions, is all the same, is regulated exactly the same way it is in human cell lines. So indeed, we've used these human cell lines to help us understand something about how this stuff is working in our frogs. What's more is one of my graduate students has shown the following. If you take a human breast cancer cell line, depends on the cell line, you can induce or upregulate aromatase by exposing that breast cancer cell line to atrazine. Here's why that's significant. Breast cancer happens when the cell goes bad. We know that the estrogen receptor, ER, is important for the promotion of breast cancer because those cells are estrogen sensitive. Now, how much sense does that make? When do you get breast cancer? Before or after menopause, typically? After menopause. So you're getting an estrogen-dependent cancer, the number one cancer in women, at a time when your estrogen levels are lower than they've ever been in your whole life, in your blood. How much sense does that make? One, it has to do with the history exposure, but the other one is we now know that aromatase is expressed in breast cancer cells, the cells around the damaged cell, so they make their own estrogen locally, which turns those cells into tumors. Now, we know so much about the importance of this so-called peripheral local estrogen production that the number one treatment for breast cancer now is a chemical called letrozole, which knocks out aromatase, decreases estrogen, so that these damaged cells don't grow and turn into tumors and spread. How much sense does that make when at the same time we're using 80 million pounds of a chemical that does exactly the opposite that we know promotes breast cancer in rats? I'll tell you how much sense it makes. It made sense to somebody because, see, Novartis Oncology offers treatments for cancers that range from breast cancer. Here's what the lawyers really want me for. The exact same company that sold us 80 million pounds of atrazine, an aromatase inducer, now sells us an aromatase blocker to treat breast cancer. And on their letrozole webpage, they talk about how this is the key to solving breast cancer. On their I Hate Tyrone webpage, they say, oh, that crazy black guy, I don't know what you're talking about. However, if you're taking letrozole to knock down aromatase and treat your breast cancer, how is that affected by the atrazine in your water, the number one contaminant of drinking water, that does just the opposite? Now I got to make clarify something. I'm not saying there's any kind of conspiracy going on. Because, see, the lawyers wrote to my dean. This guy must really have a lot of time on his hand. So I wrote a paper called The One-Stop Shop, Chemical Causes and Cures for Cancer. That got included in the president's panel on environment and cancer and went straight to Obama, which initiated this letter February 2009 from Syngenta. The lawyer said, quote, while Syngenta does share some corporate history with Novartis and AstraZeneca, it has been completely independent of both since November 2000. That's important. Syngenta simply does not manufacture or sell pharmaceutical products. Well, turns out that in January 2000, they published a paper, Novartis, saying the observed induction of aromatase, the rate-limiting enzyme in the conversion of androgens to estrogens, may be an underlying explanation for some of the reported hormonal disrupting and tumor-promoting properties of these herbicides in vivo. I didn't say that. They said that. By July 2000, a marketing application for first-line letrozole treatment of postmenopausal women was applied for. So all I'm not saying it's any kind of conspiracy. I'm just saying, boy, what an interesting set of coincidences that in January 2000, they discovered that atrazine induces aromatase and promotes breast cancer. By July 2000, they start selling aromatase blocker to treat breast cancer. And by November 2000, they stopped selling atrazine and spin off Syngenta. I bet you never in one factory where there are two people sitting at a table that went, oh, crap. It was just a coincidence, and I'm just pointing that out. So I think we're there. I think we're at the point where we can do lab and field biology and answer some important questions. We're at the point where a little boy who likes frogs can actually do something useful, which I never thought I would do in my science. But we're also at a point where I've come to another interesting place in my career. I've talked to you about prostate and mammary cancer in rats, which the company argues we can't use that to predict health effects in humans, but boy, they should have used those rats to get the thing on the market in the first place, didn't they? It causes immune failure in rats, other laboratories have shown. It causes neural damage when pregnant rats are exposed to atrazine. The EPA shows that atrazine causes abortion due to similar types of hormone imbalances that I've talked to you about. Another EPA laboratory showed that when mothers are exposed during pregnancy, their sons are born with prostate disease. Another laboratory showed that when mothers are exposed, their daughters have impaired mammary development so that their mammary glands or their breasts don't grow properly. And that same laboratory showed that when those rats give birth, their offspring suffer retarded growth and development because they don't have enough breast tissue to feed their pups. This rat is then adversely affected by atrazine that his grandmother was exposed to. This rat never saw atrazine. His mother was affected because his grandmother was exposed to atrazine during the pregnancy. And when I think about that and being useful, when I think about my sweet daughter, who's now about to turn 15, it moves me as a scientist in a certain way and as a father and as a citizen. Because this is not about you and I. We've already been exposed. Studies are showing now that atrazine, which has been banned in Europe for at least 20 years, we can't decide how long, it still has not changed in the aquifers and in the groundwater. That means that our children and probably our grandchildren will be exposed. If we take these rat studies to tell us anything about ourselves, that means that our grandchildren's grandchildren will likely be affected by atrazine and other pesticides that we're applying today. That gives me a concern, especially when I read things like this, and I will read this one out. Syngenta assumes no obligation to update forward-looking statements to reflect actual results. That's actually on their website. What disturbs me even more is that the EPA said in 2006 to the news media, the ultimate decision about whether or not to ban atrazine is much bigger than science. It weighs in public opinion. That's the head of the EPA at the time. I was advised by my advisor, don't be an advocate, let the science speak for itself. But when I read statements like that, and when I see what the company, and I love my advisor, I'm just saying I disagree. I've crossed the line, so to speak, in that I actively maintain a website where you can get information, where you can write to your congressman, where you can write to EPA and Syngenta about atrazine, and I give many public talks about contacting people so that they can do that. I've inspired and worked with Congressman Keith Ellison, who's written a bill to ban atrazine in the U.S. On Facebook now, a group of high school students and undergraduates that I've spoken to have started, and I actually get quite a bit of information. So you can join FANUS or whatever it is you do on Facebook, and there are multiple petitions for both human health-related causes and for, which I can give you if you're interested. As a result, the company has said some nasty things about me, some of you may know. They called me an environmentalist. And just so I wanted to be up on them, I looked this up, and it said any person who advocates or works to protect the air, water, animals, plants, and other natural sources from pollutions or its effects. And I thought, what scientist, what person, what citizen couldn't be an environmentalist, given that definition? But then they got even nastier. They wrote, Hayes, biologist turned activist. And I thought, for sure, they're trying to diss me now. But I checked on it. I looked it up. An activist, it says, is one that advocates or practices activism. So I looked up activism, and it says a doctrine or practice that emphasizes vigorous action. And I thought, this is starting to not sound so bad. But I looked up vigorous, and it said possessing vigor, full of physical or mental strength or active force. So I looked up active force. Active is characterized by action rather than by contemplation or speculation. That sounded pretty damn good. And a force is a strength or energy, especially of an exceptional degree, active power. And I thought, that's me. And here, I thought they were trying to insult me. Despite other advice I've been given, somebody once says, those who have the privilege to know have the duty to act. Meaning that there's no conflict in being both a scientist and an activist. He said that. Thank you. Thank you very much. And I'm not going to drink that water, but thank you for the offer anyway. There are studies with pesticides and other chemicals showing you so-called epigenetic effects or effects on methylation. I don't know of any such effects that have been shown for atrazine. In this case, the effect was as you mentioned, was the mother being affected by not having proper mammary development and then physiologically not being able to nurture properly. But I don't know of any cross-generational effects like the ones you're describing, even though I know they exist for other chemicals. There are some studies that look at intergenerational effects of DDT. One of the differences is atrazine is very water-soluble, so it's going through your body, whereas everybody in this room has DDT in their body. Every animal. In fact, we did some measurements of frogs in California, because you probably know, a fifth of the world's DDT was used in the Central Valley. And every animal there is still carrying it because it's carried in your fat and then it's passed on. The closest to what you're talking about is I know a study just came out about a year ago, and her name's escaping me, of a woman that looks at incidence of mammary cancer associated with how much exposure and how much DDT you're carrying in your body. And that exposure, as I recall, is early exposure leading to cancer much later in life, but also some cross-generational effects. Depends on what you mean. So in Europe, well, there are other triazines, some of which have been tested, which do the same thing. And it depends on your strategy, because atrazine only increases corn yield by 1.2%. So the use is an economic one, and maybe not even by that. So Europe now, there's been an analysis showing that, wait, Europe stopped using it 10 years ago. They still grow corn. Their economy didn't crash. What's the deal? So it's a very complex. That's why I was excited to hear about this group, that they're economists and scientists all working together, because it really boils down to economics and politics, not so much about, and we eat less than 2% of the corn that we grow. That's why I make that big deal out of California, that we're growing food. The Midwest is growing corn, right? That we're eating less than 2% of. During the first review in 2003, it's in part EPA in the way that we do things, I think. But in 2003, that question was asked. And a guy named Jerry White, who is president of the Triazine Network and the Kansas Corn Growers Association, said just what I heard somebody else say, that it's economic, that there's a huge economic cost to taking on the precautionary principle and doing those kinds of things. Because what happens in Europe, it's gone because if it's in the water above 0.1 parts per billion and there's a potential for harm, it's gone unless the company can prove it's safe. Whereas in the US, it's on the market unless the company can prove that it's bad. Innocent until proven guilty. So it's a very different model for evaluating those things. And they put then environmental health and safety above these economic, well, at least on the surface, supposedly, that's what they're doing in Europe. Not so much here. Oh, I hate that question. Here's why I hate that question. If I had a nickel for every Brita filter I'm sure I've sold, I'd be a rich man. Then I'd be to really talk to this. Because Brita says on the side they take out atrazine. We've actually even done experiments where we've used filters to take pesticides out of water. But I hate the question for a couple of reasons. One is it doesn't solve the environmental problem. It's still in the environment. Two is the people who are most affected aren't the people in this room. It's the agriculture workers and the factory workers, and those high levels of exposure are across the skin and through inhalation. So even if they had access to the information and the filters, they would still be at risk that way. And the other one is you're paying to take care of their problem when you do that. And even though it might, I don't know what these, but even though it might seem like it might not cost that much, there's a class action lawsuit across 6 states with water districts suing Syngenta to filter out everybody's drinking water. And the idea is that's so expensive for them that they'll stop selling the product. So it might not seem that much for you, but if everybody did it, that's a huge, huge amount of money. It's a good question. And it's not just the pesticides. We have over 80,000 chemicals that are used. And what a lot of people either don't realize or don't think about is most of these chemicals, the reason I give you guys these dates, went on the market before there was even an EPA, before 1973. So there was no regulation. It was like, hey, here's a chemical, let's use it. Many of them. So there was no testing, and especially now that we're looking at not just toxicity, but the kinds of things that I look at. Now, people always ask me, I think what you're asking, why atrazine? One, we know so much more about it than any other compound that I think it's the poster child for we need to do regulatory processes different. So it's not just about getting atrazine off the market, it's sending the message that we want these things done differently. Because a lot of people also don't realize the EPA's never banned a pesticide that was under fit for special review. So it's not like they're sticking hard on this one, it just hasn't happened. Why atrazine though? One, we have a lot of information on its adverse effects, and two, it is the most common contaminant. If you do a screen, I guarantee you at some point everybody in this room has drank atrazine. There's no other chemical that we're using today that I would be willing to say that about. Because it is the most common in rainwater, drinking water, groundwater, you name it. It is the most commonly found. Well, thank you very much.